Next, we will go through each of the four species of Plasmodium and talk about their unique characteristics. So starting with Plasmodium vivax. This one has a 48-hour life cycle completion, so it's also known as tertian malaria because invasion of new RBCs begins on the third day. This is a milder form. It invades reticulocytes usually, so the younger RBCs. And because it invades the retics, it's usually, we're seeing enlarged RBCs as the ones infected with the parasite. When you use the Giemsa stain, you can sometimes see stippling called Schuffner's stippling or Schuffner's dots. So you can see this with both vivax and ovale. And it usually contains 12 to 24 merozoites in each mature schizont. And this is one of the ways we speciate the different plasmodiums in the lab is sometimes, all the trophozoite forms look the same, and if we don't see any unique characteristics in the micro or macro gametocytes, sometimes the only way is to count the merozoites inside each schizont to be able to tell which one it is. So here's what a early trophozoite or the ring stage looks for vivax. Often you will see double chromatin dots. So the ones we looked at so far only had one chromatin dot with the small ring of cytoplasm. Sometimes you'll see two dots for vivax. And this is an enlarged RBC. This looks like it's a little broken down, but it looks like it was a trophozoite form. This is a mature troph. It's got a rather large chromatin dot, and this is an amoeboid form of the cytoplasm for the trophozoite, where it just takes on this amoeboid shape. So you'll see that description, amoeboid troph. And that's not unique for vivax, that's just another description to describe the trophs. This is a beautiful picture of a mature schizont where I can see the individual merozoites and they each contain their own ring of cytoplasm. I also see some of that brown hemozoan malarial pigment here. Now I'm showing pictures of some gametocytes, and I'll do this throughout, but usually the micro and macro gametocytes look pretty much the same among vivax, malariae, and ovale. It's falciparum that has the very unique shaped gametocytes. So for the gametocytes, for the male and female forms, you don't need to speciate vivax, malariae, and ovale. But you'll see how unique the gametocytes for falciparum look. Okay, on to malariae. This is also called quartan malaria or malarial malaria, or it's also called quartan fever because it has a 72-hour life cycle, which means the invasion of new RBCs begins on the fourth day. It tends to invade older RBCs, and because of that, the red cells aren't enlarged. They're just normal sized red cells. What's unique about malariae is that it forms band-shaped trophs. So those are often seen where the cytoplasm of the trophozoite is spread out over the whole diameter of the red cell. There's 6 to 12 merozoites for each mature schizont. And this one usually has it's the most chronic course of having malaria. It can lead to nephrotic syndrome, which you've talked about in your analysis. So nothing remarkable with this troph. It's just our typical ring form immature trophozoite. But here's what I'm talking about with that band-shaped troph. where the cytoplasm of the trophozoite is stretched out across the whole diameter. And here's your chromatin dot starting to probably multiply as well. So this is a mature troph. You'll notice these schizonts look smaller than vivax, and because of that, they tend to contain less merozoites. And the gametocytes aren't very remarkable as far as distinguishing from those other three, like I mentioned. Ovale, like vivax, has a tertian life cycle. You can also see Schüffner stippling like you can with vivax. Of the four, this is the least common to see, and it does tend to invade, well, we see enlarged RBCs, and they tend to be oval shaped, which helps with its name. And we see 6 to 12 merozoites for each mature schizont. So here's a typical ring-formed troph, immature trophozoite, nothing really remarkable. But here's a trophozoite with the oval-shaped red cell. It's enlarged and elongated, and same with these two. So this is a trophozoite. It's not a ring form because the vacuoles disappeared. It also contains Schüffner stippling, just like vivax. And I would call this a schizont because I can see individual merozoites. And this looks like a mature troph. I see cytoplasm, and it looks like this chromatin mass is starting to divide. Here are macrogametocytes. Remember I mentioned for the macrogametocytes, the chromatin mass tends to be way off center and hugging the side of the red cell. So the last one, falciparum, is the worst case of being infected with Plasmodium. It has an asynchronous life cycle, so it can have a new life cycle every 3 days or every 4 days. It's just random. You usually only see ring-formed trophs and banana-shaped gametocytes. So this is the one where you definitely need to be able to identify the gametocytes because they are so unique looking. They are crescent-shaped or banana-shaped. There are often multiple ring forms inside one RBC. It can invade any age of RBCs, and there's a wide range of merozoites that can be inside each mature schizont, 8 to 36. The patient can also have up to 50% parasitemia. So up to 50% of the red cells could be infected with malaria, which is a huge percentage. The two major things that can come with Plasmodium falciparum are cerebral malaria and what's called black water fever. So cerebral malaria is when the red cells form what's described as sticky knobs where they adhere to the capillary endothelium, and this of course causes obstructions and slows blood flow. Most notably, decreased blood flow to the brain. So this can cause seizures and altered mental status, and it can eventually cause coma and death. Black water fever refers to the severe intravascular hemolysis that can occur with falciparum. So the patient makes autoantibodies to these red cells that are infected with malaria, and it causes severe intravascular hemolysis. And remember, all that hemoglobin can get filtered at the glomerulus. And when there's so much red hemoglobin in the urine, it even looks black. Another unique thing for falciparum is called the applique form. So this is when the trophozoites hug the the rim of the red cell. So you see it's all the way on the edge here. And remember, you can often see more than one ring form inside each RBC. And this is a typical picture of a patient with Plasmodium falciparum. I see multiple ring forms. I see applique forms where they're right on the edge. Almost 50% of these red cells are infected. And I see mature schizons with the myrizoites developed. Here is your banana-shaped or crescent-shaped gametocyte. Now you won't need to differentiate macrogametocyte versus microgametocyte, but if you saw this picture, you need to identify this as the gametocyte of Plasmodium falciparum.